Hello everyone! Welcome to the Microscopy Lab for the Bio-1009 course in the Pre-Health program at Georgian College. I am Professor Maria Puibonet and will be guiding you through the main aspects of our first laboratory activity. First of all, I would like to remind you that the purpose of these lab activities is to teach students proper laboratory protocol and procedures. Therefore, students must follow guidelines and wear proper attire, such as long-sleeve lab coats, no makeup or mascara, hair must be tied back, and students should wear goggles when applicable. All teachers will have this lab set up very similarly, although there may be very slight variations between teachers. Typically, when you arrive at the lab, you will see either one set or two sets with 7 microscope stations. Except for the first station, all of the other 6 stations will be set up with a glass slide containing the specimen you will be observing and working with. Coarse and fine focus are already adjusted for you. Very minimal adjustment will be necessary for each person. Due to the number of students and time constraints, students will likely be arranged in groups of 3 students per microscope and will be given 8 to 10 minutes per station. This may also vary with each teacher. First station. Become familiar with the microscope. With the help of your diagram, locate all parts of the microscope: ocular lens, objective lens, stage knobs for moving stage east-west, north-south, coarse focus, fine focus, light switch, and diaphragm. Stations can be worked in any order except for the 3rd and 4th station, which must be done in order. We now arrive at the 2nd station. On this station, you're going to look at the orientation of a specimen and its image. A microscopic letter E is mounted on the glass slide. You will observe the orientation of the letter and how it is reflected back to you. Please follow the instructions in your lab manual. Moving on to the 3rd station. You are going to be measuring the diameter of the field of view. At this station, you will manipulate the stage micrometer. This station must be done before you do station 4. You need to know the measure of the field of view in order to be able to calculate the size of a blood cell in station 4. Station 4 now, you're going to apply what you learned in station 3. You're going to be measuring the diameter of a magnified object. This slide contains human blood smear, but don't worry, it cannot contaminate you. Using the instructions in your lab manual, you will be able to calculate the size of a single blood cell. It is very important, though, that you do not touch the coarse focus knob at this station, as it can be very difficult and time-consuming to obtain the correct focus again. Ask your teacher for help if you need it. Now we arrive at the 5th station. At this station, you're going to study depth of view. Fibers of different colors are arranged on top of each other in a crisscross fashion. At this station, You can and should manipulate the coarse focus knob so that you can move the stage up and down to give you a perspective of depth and the position of each colored fiber. Please make sure that you follow the instructions in your manual. The sixth station. At this station, we are going to apply again the concept of depth of view, but now we are also adding the concept of empty magnification. You're going to be examining a specimen called Drosophila, also known as the fruit fly. This slide contains a pair of Drosophilas. This insect is a very interesting insect to look at and an excellent way to apply the concept of depth of view and empty magnification because of its size. Please remember to read the instructions in your manual, and at this slide, avoid altering the position of the stage. We don't want to crack the slide. Station 7. At this station, you will be examining Sipha, a waterborne organism related to algae, a plant. This organism has no pigment, in other words, no color, and it has not been stained, so it is completely transparent. Therefore, it is excellent in teaching you how to manipulate light by adjusting the iris diaphragm in your microscope. Once again, I would like to remind you to follow instructions in your lab manual and review the PowerPoint slides on principles of microscopy. Make sure to take detailed notes during lab, draw what you observe, and follow proper calculations when necessary. These will be needed for when you write your report. I hope you enjoyed this video. Take care and see you in the lab.